My name is Rob Schuler, also of the Hub here, and I'll be talking about searching for face-based data specifically. So I have a short presentation and then we'll go to the site and I'll demonstrate how one goes about searching for data on FaceBase. Okay, so I'd just like to start with a couple slides just to set the context for what searching in FaceBase is like, or what kind of approach we take. And one way to describe it is to use an analogy about shopping online, maybe specifically shopping for a car online. Could be anything, a car, a jacket, whatever. It's, if you think about that, when you're shopping for something specific, you might not just use Google. Google might narrow the search to find a place to look for cars or clothing items, but you probably wouldn't find the specific item that you're looking for. And that's because the types of things that Google or other sites like Google PubMed, and so on are good for is searching for things that are primarily text-based, like documents or websites. But when you think about shopping for an item that is sort of a real-world item, it has specific attributes that you can use to locate something of interest. So if it was a car, excuse me, I'm a little congested here, but so if it was a car, you might want to know the make, the model of it that you're looking for specifically. You probably want one that's close to a certain location so that you can go take a look at it. And these are the categories or facets that classify that object. So for a car, it's like make, model, the amount of miles, maybe the cost, etc. So just looking at how would we actually go about looking for that item, and you could look at many examples. I picked one here from a popular site called cars.com. And if you go to their website on the homepage on the left-hand side figure, you don't find a Google search bar there. You find those categories, those things called facets, and it's a specific approach to navigation called faceted navigation. Now, that's the technical term, but basically it means you're filtering for an item using its characteristics. So on the left-hand side, you kind of see how that starts. The search button there is really driven by picking that item, whether it's you're looking for the Mazda and the model is a Miata. You want to find it within 20 miles of your current location, and you search. And then you get dropped into a page of results that looks like the right-hand side diagram. So over there, we have a few vehicles that have been identified. We see the filters that are currently selected, so used and Mazda, etc. And then we have more checkboxes that we can select to further refine that search. So this is the type of approach that we use in FaceBase, because when you're looking for data, the data are things about an experiment on a group of model organisms, whether they're mice or zebrafish and so on. And they have a particular age stage. There's an anatomical region of interest. There is a type of experiment, whether it was an RNA-seq experiment or a micro-CT experiment. So in the same way, these are the categories that you're looking for data. So in FaceBase, we adopted this approach. And so if you come to the FaceBase site on the homepage on the left-hand side, it has some shortcuts that are based on these categories, like mouse plus imaging or mouse plus expression data and so on, and human and zebrafish and so forth. And then when you select one of those, you go to the right-hand side and you get a listing of results. And again, you have a set of filters that you can select from, so you can further refine that search. Okay, and before I go in and demonstrate it, I just have a couple more slides to share, and then we'll get to the demo portion of this. So one of the things that I want to point out, and we don't have to spend too much time memorizing this diagram, but that there is some structure to the data in FaceBase. It is more than just, say, an abstract with files. There is some structure where we record the details of the projects that have contributed data, because that's important so that we can give proper attribution to the research projects. And these are like a project in this case is more like a RO1 investigation or a particular consortium with a specific agenda. And then these projects produce data that are collected in the form of data sets. And those are primarily what we're looking for in FaceBase, are the data sets. And then from the data sets, we have descriptions of the experiment. Experiments are described by their protocol. So we try to collect, over the years in FaceBase, it's been going on since we were established in 2008. And we have placed increasing emphasis on collecting greater details on protocol, just as the overall community has grown in terms of our standards and our appreciation for data sharing to back up scientific results. So the more recent data sets have a lot of descriptive protocols associated with them. And then those experiments are associated with the biological samples and the groups of replicates that those samples belong to. And then the data are associated with the specific replicate that they were produced from. So you know exactly where every data file was is associated with, what the biological characteristics behind the image you're looking at are, the experiment that it came from. And then finally, before switching to the demo, I do want to mention that citing data is a key part of the overall FaceBase community. So the data that you find on FaceBase are regarded as first-class scholarly works. So yes, they are associated with a publication, and they oftentimes are the data that back up the claims in the publication, but they themselves have their own value, and publishers increasingly are asking for authors in their author guidelines to recognize the data contributions that they used in their work. And so we provide on every data set page and every project page a description of how to cite that data per the leading publisher's guidelines. So we kind of follow along with Nature's guidelines and Science's guidelines about how one should cite the data. So you have all the information about the contributors to the data set. Titles are kind of optional, but we have them in FaceBase so they can be used. Of course, the FaceBase consortium itself is part of that, and then digital object identifiers that are the archive grade references to the actual data. So with that, I'm going to switch gears and we're going to look at a demonstration of the general search feature for data sets, then a specialized imaging search, and then finally, we'll look at some specific visualization examples. And with that, let me just switch my screen here. Okay, so you should be seeing the FaceBase homepage. And as I mentioned, you start off with a with the main link here over the image of the side profile view of a human face here with search all data sets. In the center, we have links here that says or get started with these links. And these are basically just preset filters that you can search on the data. So when you click on these links, they just take you to the same site, the same part of the site that allows you to search for data, but with a few common filters already set. So I'm going to go and just go to the general link without any filters set. And so this gets me the complete unfiltered set of data sets. And we can see at the top here, we're looking for data sets. We can see displaying first 25 of 1,006 records. So we can see that we have 1,006 data sets in FaceBase. And then underneath that, we have the table of results. And again, these are the first 25. And in this, we see a record ID. So these are kind of the official identifiers, or kind of like an accession number, if you're familiar with that term from other databases. The title of the data set, a summary of the type of experiments that are covered in that data set, the species, whether it's a particular model organism or whether it's human data, the age or development stages, and the PIs. In some cases, you might know exactly the researcher that you're looking for data from. And then there's a release date. Now that can be useful for just seeing what's the latest stuff, and you can sort on that, find the most recent data in FaceBase. On the left-hand side here, under the refined search, as I mentioned, the search is based on these categories that you're filtering on. So one thing I want to mention is, first of all, you'll notice there are a couple open here, but you can scroll down this list. So as I scroll down, I see the first one was experiment type, next is species, because FaceBase covers a number of animal and human subject data. We do have human subjects data. Now, if you filter on the data that are human subjects data, let's say you're interested in facial scan data, you'll find the metadata record. To actually get the data, you would have to go through a data access request process, and Alejandro will be mentioning that later. But you won't find, of course, the human subjects data just on the public site, of course, but you will find the records of interest and the ones that you can use as you're making one of those requests. So you can narrow that down here quickly. And if you're interested in going by gene or development stage anatomy, so you'll notice that there are no options underneath these. So if you click on them, you can expand each one of these categories, these filter sets here. All right, so if you're just interested in going by a particular gene, so I happen to know we have a number of data sets on, whoops, oh yeah, right, TGF-BR1, right. So we have data sets here based on a particular gene name. So you can search through, and what you see here are not just all the genes that exist, but all the genes that we have matching data sets for. So I clicked on, let me go through that a little slower. Sorry, I got a little carried away there. Okay, so on gene, you'll notice there's a show more link here. All right, so what you see here are like the first several that we have data on. You can go and directly search for things. So I could look for MSX1, but if you want to browse through the complete list, you go to show more, and then you get the complete list. Now, here's a lot of detail also on the genes, and it has not only the names of them, and these all come from the standard NCBI gene nomenclature, but the names of them, which species, because it includes a listing that goes across species, the description, and additional synonyms. And of course, there can be quite a few synonyms for each of the gene names. And you can search through those, and we can see we have data sets that cover over 516, well, not over, 516 genes. So these are all, there's available data on any of these in the FaceBase site. And one thing I want to point out, is that any of these can be expanded. You can find filters in a number of different categories. And one thing that there's sometimes a bit of confusion about is the feeling that maybe I have to start with experiment type and then filter my way down. I might have to, as if you have to start with, say, RNA-seq assays and then filter the next. And I just want to say that there's no hierarchy here. Just like in if you were shopping, there's not really a hierarchy. You might have to say, okay, I'm looking for, if I was on Amazon, I might have to say I'm looking for shoes or jackets or something like that. But but otherwise there's no filter. I don't have to pick price before I pick color, for example. It's not a hierarchy. So in the same way on FaceBase, you can filter based on any of these categories if you're just interested in a particular data on anything that we have on mandible. So I go looking for mandible, I find it here on the list, and I filter on that. And so I see I have 19 matches, and now I can go from here. I can browse through and find the data set that I'm interested in, and you can continue to further refine that. So let me just pick mandible and mandibular process. Let me go up. I would also mention that all of the filters are being updated as I select on a particular option. So when I picked mandible and mandibular process, the genes were reduced there. So now let me go back and click show more. So I went from 516 to now 29 records. So I'm able to keep refining my search down, my search results down to a specific item that I'm interested in. All right, so if I go back up here and I say, well, let's see what's the single cell data I have. So now I've got a filter and I can see it summarized at the top here. So under data set and then the general search bar, I have a little a breadcrumb here in a way for the experiment type being set to single cell, RNA-seq assay, and the anatomy filter being set to mandible and mandibular prominence. And I end up with 3 search results available to me. So this is basically how one goes through looking for the data on the FaceBase site in general for data sets. Let's see, I'm going to open up another tab here and go back to FaceBase. Okay, so I'm going to start over here on the FaceBase site because I wanted to keep my place there, but show you another search avenue in FaceBase. So another one is the image search. So from the home page here, I have a nav bar, data browser, and then images. So if I really just want to dive right into the images, I can go to this data browser images site. And again, we will see an interface that is functionally similar to what we just looked at, but this goes directly to all images. So we have 45,000 image records in FaceBase. Now, some of the imaging data are not all pictures. Some of them are landmark data, and some might also be some amount of metadata. So out of those 45,000, maybe, I forget exactly the number, but some fraction of them, maybe a third are landmark data or something about like that. So I can see here initially I have a variety of TIFF files. You can also filter out the ones that maybe you don't want to search on. So for one thing, we have the experiment type. Now with FaceBase, all of the data, I showed you that one figure where we had the project and data set. I talked about the experiments and finally the data at the bottom. So all of the images, these fit into that little blue box at the very bottom called data. Now they're all linked into all of the rich information that we have in FaceBase. So the search over the images can be driven by all of the same features that drove the last search for data sets. So we can go by experiment type and species and anatomy and stage and so forth. So we could look for things that are fluorescence microscopy, for example, and see what results we have there. We could look for, let's see, micro CT. So if we were just interested in micro CT images. All right, we probably have a lot of results for that. All right, oh, it's got a update some filters too as we go along. Oh, all right, I think that we have a little issue there. All right, we'll have to look into that later. All right, that's probably because the result set size made it not too happy there. But let's kind of go to confocal fluorescence microscopy. So we can search through based on these filters, and then we can also reduce our space with species, like if we were interested in just zebrafish images. So we can go through the data and then drill down into a particular image. Let's say we can pick one of these images here. We can find the data set that it's from. You can see a little closer up image of the thumbnail, and we can actually find our way back to, say, the data set that it came from. We found something of interest and we want to learn more about it. So we can get the complete description of it. We get details of the kind of the high level keywords associated with it. We can see all of the complementary data, other images in this case. We get the description of the experiments here, and then all of the biosamples and so forth in terms of the data that are associated with that image and the broader set that it comes from. Something that I mentioned in the slides that I wanted to point out again on the site is that as you get to a data set, and like, let's say you found images of interest, you use them, you're able to have examples of how to cite them. So in the upper right-hand corner, almost near the upper right, is share and cite. So if I click on that, I get the share and cite pop up. It gives me a couple things at first. You'll notice a versioned link. This is kind of, will always give you this exact snapshot, the shot of this data set. And this will always be static, kind of a fixed time point view of this data set. Even if something changes in it at all, like a description is updated, this will always be consistent to what you saw when you were browsing the site. Then there's the live link that will be updated if the data set were to be updated. That doesn't usually happen after a data set is initially released, but occasionally there might be a revision to something within a month or 2 or 3. But then beyond that, usually the data sets aren't going to change after that. And then you'll see the data citation example given. So this tells you how you would put this into your reference manager, how you would refer to the data that you used on FaceBase. And then finally, there's a format. You may or may not be familiar with it, but it's a standard format that can be imported into EndNote or any number of other reference managers. And it's called BibTeX, and that'll have all of the details to be able to reference this data set. Okay, so with that, I just wanted to just take a look at a few examples of the visualization features, some of the newer capabilities on the site. Well, some existing, and then a couple newer ones. So let me just kind of go through those before I hand it over to our next session. Let's see. So I picked one particular data set here, and again, using the search interface, I could go through and find them, but just in the interest of time, I have them picked out already. But essentially, we can see up here what these are about. So it's mouse data, MSX1. We have E16.5. We have its genetic background. We have the strain that it came that they were sourced from. Okay, and so the data set includes a top level thumbnails. We can get a quick idea of the data that's in here. And then we have a section of downsampled image previews. So if we were to download the data, we would find some full resolution NII, which is nifty format micro CT images. So nifty is one particular medical imaging standard, and it's pretty widely supported. So if you were to use an open source tool like Fiji or ImageJ or most commercial tools, you would be able to open this data. So on the site, we have the data also visible. And what you'll find is that you can, let me just show you, start by click here to load the image. I did that just so it would be preloaded here, and we could skip that step. But you begin with this orthogonal slice viewer. And so we have the various panes here. So I can slice through the sagittal pane if I like. You can also adjust some of the threshold for what gets displayed. And then there's a feature here that can easily be missed, but in this image window in the left-hand side, left top, there was a little label here, volume. I'll kind of move away from it again. So as I get closer, it'll pop out here. And if I click on the 3D button in there, what'll happen is this image, which we're currently looking at in ortho slice, gets converted into a 3D volume. Now I mentioned that you have to do some adjusting of the threshold. So in this case, it's not too interesting if we don't make that adjustment. So if I come over to the threshold bar and move that, I can start to reveal the image. Occasionally this is needed because just because of the default settings of the image, and we try not to adjust that in a permanent way. So we leave it up to the user to play around with. So as I adjust that, now I see kind of an image is displayed, and I get a good view of the specimen. All right, so this is our orthogonal slice viewer and volume image viewer. Now the next example, I think I'm going to have one more example of that because I wanted to show a slightly different kind of data. So some of our newer data we have broadened our collection to include some dental data. So here's another such viewer, but with a different set of data. And we can kind of go in here and again, convert this to 3D. Okay, and in this case, without making any threshold adjustments, it was viewable as is, so I won't adjust anything. And it does do this little processing step a couple times before it's completely done, because it'll do it as you rotate around a different axis, it'll process again. But we can see some images online here, and you can of course download this, get the full resolution image for your own offline viewing. Now, if I go down in this data set, keep going down, again, we have the description of the experiments. Okay, these are, we can see it was micro CT, and there's also H&E staining experiments. We see the biosamples. We can get a quick idea of the strain, and here's a range of age stages. So this looks like it's all postnatal data. And as we go down, we have images in a variety of formats. We have some gross anatomy, probably CT data, followed by the H&E histology data, and so forth. Now, I have a particular image picked out here, so let me switch here. Okay, here we go. So here's one of the images, one of the histology images, and what we wanted to point out is that we have a new feature that we are working on getting more data in this and enhanced with these image annotations. So I'm looking at one of our microscopy images here, and I can see that I have some little figures or annotations drawn on it to pick out particular regions of the image to point out things that are of interest. And you can see I can zoom in and pan around. Our viewer actually supports very high resolution imaging. So really whatever is contributed to FaceBase, we can handle very high resolution images, and they can be annotated. And those annotations are linked in with the anatomy terms and so forth on the site. All right, and with that, let's see, I have I believe, one last visualization example. Okay, and now this is, let me just scroll to the top of the data set. I'll start from the top here. This is for a single cell RNA-seq experiment. And we have a data set here that was contributed, has a couple experiments. Again, this is actually chick data. So we have Gallus. And that's also kind of a new thing to FaceBase. So we are increasingly encouraging additional, broader types of data being contributed to FaceBase on other relevant model organisms for craniofacial, oral, and dental data. And another new feature here is the single cell viewer. So if I see that I have a thumbnail here on one of the processed data images, this gives me an idea here. Okay, it's R data. So if you're familiar with this, R data is a particular data format, general data format used in R, and in this case, it's a Seurat object. So this is a particular interest to single cell visualization. So let's see if I open that up. Okay, and what we get here is a quick view of our the use of our integrated UCSC cell browser. So this comes from Ucsc. So if you're familiar with their tool, or you want to learn more about how to use their tool to browse and visualize single cell data, you can go to the Ucsc site for the cell browser. And that's what we use on FaceBase. So we have a few data sets that we now have online visualization for, and we are in the process of enhancing our single cell visualization pipeline to handle additional data. So more of our single cell data will show up for online visualization. And then, of course, more of the data that's coming into FaceBase tend to also be single cell visualization data as well, or single cell data, single cell experiments. So more of this data will be showing up on FaceBase. So with that, this concludes my part of the demonstration, and I will hand it back over to Chris.